Hello guys, let's begin with another lecture today. Today we'll talk about the membrane protein. We know that the two kind of membrane protein which can be present on the membrane or the plasma membrane or any kind of membrane. It would be the extrinsic protein, which is also called as a peripheral protein. Another one is the intrinsic protein, which could be the embedded one or it could be the transmembrane protein. Let's deal this protein with another way. Here we'll talk about the four different kind of protein. One is a transmembrane protein, which is a kind of integral protein, which can span the membrane from one side to another. Another protein is the cytosolic membrane protein, which is present towards the cytosolic end of the membrane. Another one, the external membrane protein, which is found outside. And these two can be integral or can be the external protein as well. And then we have the peripheral proteins, which are loosely attached to the membrane. So this is how it is going to be. Now there'll be few things we'll talk about it. One, that basically the peripheral proteins, they can be present on the either side of the membrane and they're going to be associated with a bond or the polar end of the transmembrane proteins by a non-covalent bond, or it could be the ionic interactions, which is very loosely attached, which can be easily removed. You can see over here they may have the other bonds to also be formed. You can see some of them, they can be ionic ionically ion bonds, and some of them you can see they're bound to the part of the membrane with the covalent bond. You can see over here these are the non-covalent associations with the polar end of the transmembrane protein, and the second, which is shown over here, that could be the covalent bond. But important question is that how the transmembrane protein cross the bilayer. This is very interesting question to answer. Why it is very important is that the whenever a protein has to cross, it has to cross the lipid part, which is highly hydrophobic. So any protein which is going to pass through it must be containing the hydrophobicity. Otherwise, it will not be able to cross. It will not be able to maintain its integrity during the structure of the plasma membrane. So most of them they do in the alpha helical form. Why we'll talk about in a little while. So this alpha helic composed largely of the hydrophobic amino acid. The propensity of the amino acid which is found in the alpha helix is highly hydrophobic amino acids. Okay, so that helps them to pass through the lipid layer, which is hydrophobic regions. You can see over here. So you can see the residues, which is highly hydrophobic. So this is the alpha helical structure, which allow them to pass through the one part of the membrane to the other. This span the membrane from extrasolic cytosolic region to the cytosolic region. Now, one example of this is the glycophorenes, which is having, you can see over here, this is the cytosolic region and this is exterior of the cell. And this is the alpha helical region, which is present in the lipid bilayer regions. And if you see, this is a single pass protein because it is crossing the membrane only once. Then if you talk about the peptide bond, that is going to be polar. And in this alpha helical structure, there will be no water because of the hydrogen bond formation. You can see over here, carbon, nitrogen, hydrogen. We know the hydrogen bond will always be formed between this oxygen and this is the the hydrogen, which is present over here. So that is present on the nitrogen part. So NH is the peptide bond and the hydrogen bond to the cytosolic regions, basically this I+4 structure. So every 4 amino acid will have the bond, and you can see the distance here is going to be 0.54, which can help you in solving the many problem in the biochemistry. And this is the carbonyl group. Other protein that transfers the bilayer as an antiparallel beta structure. In the biological system, we have the more antiparallel beta than the normal parallel beta, because antiparallel is more stable and the parallel beta structure is strained, hence the stability is going to be less. And many of them, they form the beta barrel. And you can see over here, this could be the 2 layers, or there could be more than 2 layers. And you can see the orientation is going to be in the opposite direction. And they form the interchain hydrogen bond. And this number of the chain can be more than one, or more than two. So especially beta barrel proteins, they form the transmembrane channel, especially all the outer membranes of the bacteria or the mitochondria or the chloroplast, because of porins. You just have a beta sheet and you can just roll over. So that such kind of water-filled pores can be formed. These are called as porine barrels. So usually they have the 16 standard anti-parallel sheet, which is rolled over, and they make a cylindrical structure. You can see over here. So here, if you see the polar amino acid residues lying in the aqueous channel, which is inside, and the outer part of this is going to see the non-polar amino acid, which is directly interacting with the lipid part. So one way to ascertain this is the hydropathic plant. So usually the membrane proteins are difficult to get crystallized because they have a lot of hydrophobicity. And once you go for the X-ray crystallography structure, which is the one of the way to study these hydrophobic proteins, what you'll come to know. And another way is the DNA cloning. And what you come to know that, the region which is going to be in the lipid part is going to be hydrophobic. And sequencing can also be done. And that is how we going to, come across with the region, especially this 20 to 30 amino acid, which is having the very high degree of the hydrophobicity. These are present, in the lipid part. Otherwise, the structure is going to be unstable. Thermodynamically, that structure will not be feasible. You can see over here. So this is the large peak. The value is going to be more than 0. Here, you can see there's a 1 peak. If you come across, that means the protein is going to be single pass. And if the peaks are 7 over here, so this is going to be 7 pass protein. This is what the hydropathy index will show you. And those peaks which is below 0, that shows you these are the regions which are having the hydrophilicity. Now the solubilization of the protein for the study purpose, you require a detergent. What is a detergent? Which is an amphipathic molecule having both the regions, polar and non-polar. The polar part will attach to the polar region of the membrane protein, and non-polar will bind with the non-polar region. It will drag into the water, make it solubilize. So once you put this, it is going to show you this micelles where the head regions are on the outer part, which is directly in the contact with the water, and the tail happen to be the hydrophobic, which is going to be lying into the center. So if you see this, let's say the one protein is here, and the dark green part is showing these are the hydrophobic regions which is coming in the contact with this. And once you add this detergent molecule, they make the micelles. And look at over here, the tail region is going to bind over here, and some of the lipid part as well, and they will drag them into the water soluble protein lipid detergent complex, and the lipid will be dragged out of the soluble mix with the lipid detergent micelle. You can see over here. So these are the very strong, especially the peripheral proteins are very easy to be removed, but the integral proteins are going to be difficult. So you require the strong detergents to be use them so that you can analyze them. Look at over here. Can we again just look at over here. This is your sodium potassium ATPase pump. Once you treat them with a detergent, you can see this protein is going to be soluble. Here is going to be lipid. You can purify the sodium potassium pump, and if you want to reestablish, reconstitute it, so you can remove the detergent and put into a lipid. So what is going to happen? They're going to make this structure in the proper orientation. So this is how the reconstitution is going to be done. These are the few detergent SDS. You can see over here, it's a negative charge over here, and you can see the non-charge. And this is another one, the octyl glucoside. These are the detergent having both the regions, hydrophilic and the hydrophobic regions. And some of the membrane protein you can see over here, They can make the amide linkage, they can make the thioester regions, they can directly, bound to the lipid part. So membrane protein attached to the fatty acid chain or prenyl group. You can see over here. So the thioester or the thioether linkage can be the three kind of linkages that you can see over here between the proteins which are bound to the lipid part. And one is going to be the amide linkage, or it could be the thioester, or it could be the thioether linkages. So proteins that function only one side of the lipid, by contrast, are often associated exclusively either by the lipid monolayer or protein domain on that side, and they play a very, very important role. You can see over here. Suppose we have the mouse cell and we have the human cell. You can see that they vary in the protein. Let's go for the fusion, and we created the heterokaryons. So what we'll do, we raise the antibodies both for the mouse as well as with the human proteins, and we tag them with the different kind of the fluorochromes. Like in the case of here, we use the fluorescein, and here we use the rhodamine. One is a green in color, another one is the red in color. With the time, what we'll find is that they have the mix of both. Here you can see you can easily separate them out, but it is a mixed one that suggests the protein do have the some diffusion activity across the membrane. Another way of showing the same kind of phenomenon, the two techniques which could be used, one is a FRAP, another one is a flip. So FRAP stand for the fluorescent recovery after the photobleaching, and flip stand for the fluorescence loss in the photobleaching. Look at over here. So in the FRAP, what exactly we do, we bleach the certain area by giving the laser beams in the beginning, and then what we'll do is that we give them some recovery time. So what happens is that these regions are going to be now little lightened up, but the bleach area is going to be entirely covered. So this graph shows here the bleached area, and after that the recovery. Recovery means that the mixing is going to take place, so that you have the color development in the either part. This also suggests that the protein, they do diffuse along the membrane. Another technique is a flip, fluorescent loss in the photobleaching. What we'll do over here is that we measure the area and bleach with the laser region, and we start continuously bleaching there, and this area keeps on getting mixed up, and finally we get the area which is the lighter in color. A time will come after the continuous bleaching, the entire region become they will lose the fluorochrome they are attached with. That also suggests the protein diffuse. So flip, FRAP, these are two techniques to ascertain that the diffusion of the protein do takes place. Now, the certain proteins shown over here in the RBCs, you can see the band 4.1, tropomycin, spectrin. So these proteins, they do play important role in shaping of the cell. Now, the another important thing is how to study the sidedness, whether the particular part of the membrane is inside or the outside. So the easiest way to work this out is on the RBC, because it does not have any kind of the cell organelle present over here. So the ghost cell will be used for the purpose. Look at over here. What we'll do is that we'll take the RBC, we put into the hypotonic solution, it will get burst, we wash it, and we'll have the leaky ghost, and we allow them to reseal, or we can disrupt this again, reseal. So what you see over here is the red color that's shown outside. It can be right side out, or it can be the inside out. Okay, so inside out means the inner part has gone onto the outside, and the outer part has gone to the inside. Right side out is the same that we have in the RBC, but they are broken to the small vesicle form. Okay. Now, how to study the protein? So sidedness of the protein can be determined by the vectorial labeling. We use a covalent reagent that could be the radioactive, or it can be fluorescent, that binds to the protein of the protein exposed to the membrane surface. Reagent needs to be water soluble, so it won't permeate the seal, because if it is hydrophilic, it will not go inside the cell or inside the vesicle form. For example, label seed right side out vesicle, we separate them with the SDS phase. Label protein detected by their radioactivity or their fluorescence. If it is on the outer side, it will fluoresce or it will show you the reactivity. You can see over here. So SDS page result like the alpha spectrin, beta spectrin, and chirine in the right side out vesicle. So A part you can see over here, the gel is stained with the comessive blue, and B side is drawing the position of the some major protein. You can see over here glycophorin labeled, indicating it is exposed on the outer surface. Now we'll go into the inside out. So the again we'll do the same thing. Here the gel is going to be stained with the comessive blue, and B part drawing the positioning of the some measure. So here what happened is the glycophorin label indicating it is exposed on the inner surface of the membrane as well. That means it is going to be a transmembrane protein. And this is how it looks like. So this is the exposed part of the outside. This is the exposed part of the cytosolic region. And major plasma membrane proteins of the red blood cells, we can establish there will be six major proteins which could be present, and this is shown in this SDS page diagram. So spectrin is one, which is a peripheral protein on the cytosolic region. A long, thin, flexible rod, which is a principal component of the cytoskeleton, which is underlying the surface of the red cell, spectrin. Then it has the role to play, the maintenance of the structural integrity, because it is having a role to play with the the cytoskeleton, which determine the shape. Okay, and if you go into the detail, what you'll find is that it is a heterodimer of the two antiparallel polypeptide chain termed as the alpha and beta. Heterodimer self-associate head to head at the phosphorylated part you can see. So electron microscope of the isolated spectrum you can see over here. Then we have the tetramers linked together 4.1 by the actin. So this form a junctional complex and serve as a deformable protein mesh, allowing the red cell to withstand stress when it is forced through the small capillaries. So this is all about the different proteins which are present on the RBC. Hopefully you like this presentation, and I will see you in the next lecture, which will come soon. And keep on watching, sharing, and subscribing my channel. You'll get more in the future. Thanks a lot for the lectures.